Chapter 8. Cell Structure and Functions. Cell Structure and Functions. Though non-living objects may perform one or more of these processes, like vehicles can move, but it is only a living thing that perform and exhibit all the above characteristics. Amongst the living organisms, these are broadly classified into plants and animals. All these organisms look different from each other. However, all of them are made up of microscopic units called cells. Discovery and history of the cell. An English scientist, Robert Hooke, observed slices of cork under his self-designed crude microscope. Cork is a part of the bark of a tree. He took thin slices of cork and observed them under the crude microscope. He noticed a large number of compartments in the cork slice, resembling the structure of a honeycomb. Robert Hooke coined the term cell for each box, which in Latin means little room. Unicellular and multicellular organisms. Living organisms are made up of one or more cells. Organisms made up of a single cell are called unicellular organisms. Amoeba, Paramecium, Euglena, bacteria and yeast are few examples of unicellular organisms. All the basic functions of a living being, like respiration, excretion and reproduction, are performed by one cell. Some unicellular organisms. Organisms made up of many cells are called multicellular organisms. Most of the plants and animals that we see around us are multicellular organisms. Trees, grass, insect, cows, birds and humans are few examples of multicellular organisms. Cell, shape and size. Cell shape. Cells show a great variation in their shapes, such as spherical, eggs of many animals, spindle-shaped, smooth muscle fiber, elongated, nerve cells, oval-shaped, red blood corpuscles, branched, osteocytes, and so on. Some cells may not have definite shape. Amoeba, white blood cells. Cell size. The cells and their organelles are measured by fractions of a millimeter mm. 1 micron or micrometer equals 10 raised to the power minus 3 millimeters. The size of cells varies from the very small cells of bacteria, 0.1 to 0.5 micrometer, to the very large eggs of the ostrich, 170 mm into 130 mm. Structure of a cell. The cells differ in their shapes and sizes. The animal and plant cells are not exactly the same. However, they show some similarities in their structure. A typical cell consists of 3 parts: cell membrane, cytoplasm and nucleus. Let us study each in detail. Cell membrane or plasma membrane. Every cell is bound by a thin delicate membrane called cell membrane or plasma membrane. The membrane separates cells from one another and from the surrounding medium. Functions. Plasma membrane is porous and allows the entry and exit of only selected substances. It also prevents the movement of some other substances across it. Thus, it is also called selectively permeable membrane. It provides an outer boundary to the cell and protects the cell from injury. Cytoplasm is a thick, jelly-like fluid inside the cell membrane. It occupies the space between the cell membrane and the nucleus. There are many small cytoplasmic bodies in the cytoplasm.
Each community is marginalized for diverse reasons and in different ways. We all need to play an active
These are called cell organelles.
enshrined in the constitution in order to preserve the diversity of
Nucleus. It is the most important component of the living cell. It is generally spherical and located in the center of the cell. The nucleus consists of the following four parts. Nuclear membrane. Nucleus is separated from the cytoplasm by a double membrane called the nuclear membrane. This membrane is also porous and allows the movement of selected materials between the cytoplasm and the nucleus. Nucleoplasm. A dense fluid in the nucleus is called nucleoplasm. Nucleolus. It is a small spherical body present inside the nucleus. Chromosomes. Chromatin material is thin, thread-like structure composed of deoxyribonucleic acid, DNA. This contains genes. Genes are responsible for passing genetic characters from parents to the offsprings. During cell division, the chromatin material condenses to form thick, ribbon-like structures known as chromosomes. Functions. The nucleus controls all metabolic activities of the cell. It is responsible for passing genetic characters from parents to the offsprings. That is why the offspring resembles their parents. If a person has brown eyes, his son or daughter may also have brown eyes. However, the different combination of genes from parents results in different characteristics.
Objective: to prepare stained, temporary mount of onion peel cell and to record observations and draw labeled diagrams. Click on the things needed. Click on the onion to place it on the table. Click on the knife to cut the onion into pieces. Click on the watch glass to put a piece of onion on it. Click on water to submerge the onion piece in water. Click on the forceps to remove epithelial membrane from the onion. Click on the blade to cut the peel into a square or rectangular shape. Click on watch glass again. Now, there are two more watch glasses, one for safranin. Now, click on the safranin to put it over the watch glass. Click on distilled water to put it on another watch glass. Click on the glass slide to put it between the watch glasses. For transferring of the, click on the brush to shift the peel to safranin solution to stain the peel. Then to watch glass with water to remove excess stain, and finally to the glass slide. Click on glycerin to put a drop over the peel kept on the slide to fix it on the slide. Click on the needle to help putting the cover slip on the slide. The needle is used now. Click on the cover slip to cover the peel. Click on the blotting paper to remove excess glycerin from the slide. Click on the slide to place it on the microscope. Observation. You observe a large number of rectangular cells lying side by side in the epithelial membrane. Each cell has a distinct cell wall. Intercellular spaces are absent. A distinct nucleus is present in each cell with a prominent large vacuole. Conclusion. The cells observed are plant cells, as each cell consists of a cell wall and a large vacuole. Precautions. 1. Always use a brush to transfer the onion peel. 2. Avoid entry of any air bubble under the cover slip. 3. Avoid overstaining and understaining of the peel. 4. Avoid keeping the peel too long in water after staining. 5. The cover slip should be gently held with the help of a needle. 6. Remove extra glycerin from the slide with the help of a blotting paper. 7. Always mount the specimen in the center of the slide.